Okay, this is chapter 21 and it addresses the immune system, and we're going to discuss all the components of the immune system, how it protects us against disease, and where the important structures are located. And we'll talk about the different types of defense, including um, innate immunity, acquired immunity, and um, the use of what we call our barrier functions in order to keep us healthy most of the time. Okay. We'll also talk about where the immune system comes from and how it gets established, and how it determines whether or not something is a threat to the body or not. We have different systems that are designed to protect us against disease because we live in a world that's filled with stuff that tries to make us sick, and these things are called pathogens. Okay? Pathogens can be living or non-living. Living pathogens include bacteria, mold, or you could say fungus, parasites, right? Even in some cases, insects, right? Or protozoa in some cases. Now, some pathogens are not living, and those include viruses and toxins. Okay, but the point is, they're around us, they're in us, they're on us all the time. And if we didn't have a way to defend ourselves against disease, we would constantly be sick. So, when we talk about non-specific immunity, we're talking about a body system that uses the same approach regardless of the nature of the pathogen. Our first line of defense includes our mechanical and our chemical barriers. Now, what are those? Okay, our mechanical barriers are our skin and our mucous membranes. Okay, and the presence of things like the hairs in the nose, the vibrissae, that act as a filtering mechanism. The chemical barriers are the secretions that come from those mechanical barriers, and they include mucus from the mucous membranes, oil and sweat that comes from the skin. Okay, and there are components in those secretions that retard bacterial growth. Okay, there are antimicrobial compounds, and there's also an acidic pH that's produced on the skin especially, as well as a high salt content that tend to retard growth. And the fact that the upper layers of the skin are constantly shed and remade also cuts down on the ability of the skin to be colonized. Okay, our tears, our saliva, our sweat also contain things like lysozyme, which weakens bacterial cell walls. Meganins are components that are found in ocular secretions, and they punch holes in bacterial membranes and make it easier for them to basically be killed when they have an osmotic shock, and causes them to rupture. Okay, but all of this is going to use the same approach regardless of the identity of the pathogen. The next line of defense in the event that our mechanical barriers are breached includes phagocytosis, which is the ability of certain cells in the immune system to eat pathogens through essentially consuming them and forming a little vesicle around the pathogen, and then inside the cell, lysosomes fuse with that vesicle and break up whatever's inside it, and that destroys it. Okay? The inflammatory process, which we'll learn a little bit about in a minute, which is essentially where an infected or an injured area turns red, swells up, warms up, and hurts. Okay, and that's designed to bring blood to the area, and with the blood come the white blood cells, and that helps protect against disease. Fever, which creates conditions that pathogens don't like, but which our white blood cells do. and it makes it easier for us to kill them. Okay. There are proteins that are going to defend against our pathogens as well. These are contained primarily in the vesicles of granulocytes, which are basophils, neutrophils, and eosinophils, and they essentially either punch holes in membranes of pathogens and cause their cytoplasm to leak out and they die, or they induce cellular suicide called apoptosis. Okay, and then natural killer cells that basically punch holes in either infected cells or cancer cells and cause the cytoplasm to leak out and they die. But in every case, these chemical weapons are not specific for the pathogen. They are going to use the same approach regardless of whether it's a bacteria or a parasite or what have you. Okay. The third line of defense is different. The third line of defense is based on the presence of molecules on the surface of the invader called antigens, which are specific for that invader. And we generate then a team of white blood cells called lymphocytes that are only going to go after whatever the antigen is attached to. Okay, if it's attached to an infected cell, it's going to kill the infected cell. If it's attached to the pathogen, it's going to try to kill the pathogen. And there's a variety of techniques that it uses to do that. Okay, so you can see here all the different components of immunity that keep us relatively safe all the time. The digestive system is kind of interesting. If we look at the back of the throat, we have tonsils, which are basically collections of lymphocytes and macrophages, and they trap ingested pathogens. Okay, the mucous membrane that lines the entire digestive system from the end of the esophagus to the beginning of the rectum is going to produce mucus that flushes pathogens out and traps them and keeps the pathogens from penetrating through and getting into the blood. And then the fact that the stomach is an acid bath that sterilizes the food that we eat is another way that we again cut down on infection. And we should also point out that in the layer of tissue under the epithelium, the mucous membrane that lines the digestive system, are lots and lots of white blood cells that are going to defend against anything that gets through the epithelial layer and tries to get into the bloodstream. Okay, so that's just an example of how really all of these systems are present in the digestive system. The point being that again, you need all of these acting together in order to keep us from getting sicker quicker. Okay, so let's look at phagocytosis, right? Essentially what happens when you injure or infect tissue is that there are chemical signals given off that draw white blood cells to that area, and then the white blood cells are going to start by eating pathogens and cellular debris. Okay, so let's take a look at how this happens.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation.